Welcome back. In today's class, we will be learning about eukaryotic organisms and its ultrastructure. Salient features of eukaryotic organisms are its ability to have the membrane bound organelles. Eukaryotic organisms particularly have nucleolus has been covered with the nuclear membrane. They have many membrane bound organelles. The best example would be nucleus that is enclosed within the nuclear membrane. Eukaryotic organisms are quite complex or evolved when compared with prokaryotic. Eukaryotic organisms include protozoan, fungi, plants, and animals. Eukaryotic organisms are generally having certain membrane bound organelles, and we're going to divide the membrane bound organelles as single membrane bound organelles and double membrane bound organelles. They are the ones who have mitochondria. They are motile in nature. They have a typical cell wall layer. They show complex reproduction, and the genetic material is linear DNA. When we speak about the ultrastructure of a typical eukaryotic cell, this is how it looks, starting with the nucleus. Nucleus has been divided into various components such as the nuclear membrane or the nuclear envelope. As we saw, we know that nucleus is enclosed inside the nuclear membrane, also called as the nuclear envelope. Inside that is a nucleoplasm, which is a space where the genetic material is present. Genetic material in eukaryotes are linear DNA. We know that it's DNA. It is also called as chromatin. And nucleolus is a part of the nucleus which is highly pigmented, and we will get back to its function. Now your nucleus is always attached with the endoplasmic reticulum. Endoplasmic reticulum is divided as smooth endoplasmic reticulum and rough endoplasmic reticulum. Rough endoplasmic reticulum will have show that beaded structures or bulgings, those are nothing but the ribosomes that are attached to the endoplasmic reticulum. Because of this bumpy appearance, they are called as the rough endoplasmic reticulum. Smooth endoplasmic reticulum do not have ribosomes bound to it, and they appear smooth and continuous. That is why we call it a smooth endoplasmic reticulum. We have ribosomes, we have intermediate filaments, microfilaments or microtubules. We'll come back to its function. And cytoplasm is a house or a habitat where all the organelles are present. We have peroxisomes, yes, we have mitochondria, we have vacuoles, we have Golgi apparatus, and we will be looking at all the functions of this. So as I told you, these organelles are present in the eukaryotic cell, and we're gonna divide them as single membrane organelle and double membrane organelle in the upcoming videos. Foremost difference between your eukaryotic cell and a prokaryotic cell is the having of your membrane bound organelles by your eukaryotic cell. They are quite evolved when compared to prokaryotes. They show complex metabolism, complex motility, complex reproduction. So under your eukaryotes, we have protista, we have fungi, we have animalia, we have plantae. All this comes under your eukaryotes. So the cell wall of your eukaryotic cell is also called as the cell membrane or your plasma membrane, made up of three important components called proteins, carbohydrates, and phospholipids. Proteins, carbohydrates, and phospholipids are present in the cell wall of your eukaryotic cell. Here, the cell is believed to be hydrophobic or hydrophilic in nature. The cell wall is having two components, that is hydrophobic and hydrophilic. That is your phospholipids. Phospholipids are nothing but your lipid with phosphate group. When we try to decode that, they behave or are observed as the head and the tail. Head is believed to be hydrophilic in nature, present towards the environment where there is water molecules present. We know hydra is water and philic is loving. So hydrophilic end head is to approaching towards the environment, and it also has a hydrophobic tail which is present sandwiched between the two heads, that is your hydrophilic environment, hydrophilic head. So there is, it is cell wall is always a double layered. Towards the outside there is head because it is hydrophilic, and then to sandwiching the tails between the heads, that is your hydrophobic tails. The function of your cell membrane is its permeability. It is semi-permeable in nature, allows only selective molecules to pass through it. They are capable of withstanding the shock, protect itself from the external injuries. Here, these molecules will also have certain receptors to interact with the molecules in the environment. They also play a vital role in segregation of the cell growth, or division. So they show two types of permeability, and they show two types of transport, that is active transport and passive transport. In active transport, transport occurs against the concentration gradient. It requires vital energy. Protein carriers are involved to transport the molecules from the environment to within the cell, and they are highly selective in nature. But whereas passive transport is along with the concentration gradient, they do not require any additional energy. Protein carriers are not involved, and it is not highly selective. So coming to the cytoplasm behaves as a house for the organelles to reside. It is made up of a semi-solid or jelly-like structure, hydrated. It is capable of supplying all the mineral nutrients to the organelles present there. That is also called as cytosol, which is a water-based solution, contains many ions, small molecules, and macro elements in that, in which all the organelles of the cells are present. These ions or small molecules or macromolecules include macromolecules such as glucose and simple sugars, polysaccharides, amino acids, nucleic acids, fatty acids, or ions such as potassium, calcium, and other minerals that is required for nourishing the organelles present there. Cytoplasmic matrix can also be called as the cytoskeleton. This creates the ultrastructure, or the contributes to the morphology of the cell. When the cell is long, linear, it is because of the cytoskeleton. Skeletal structure of your cytoplasmic membrane. This not only contributes to the shape or the internal organization, it also provides mechanical support to the cell, but also helps in division and motility. These cytoskeletons are nothing but made up of filamentous protein. There are three types of filamentous proteins. That is, your cytoskeleton is made up of three types of filamentous protein. The first one is microtubule. This is how exactly your microtubule looks like. It is made up of tubulin protein. Microtubules are made up of tubulin protein. This is the largest type of filament present in a eukaryotic cell, that is about 25 nanometer in size, and they are composed or made up of tubulin protein. Microtubules are made up of tubulin protein. There are two types of tubulin, alpha and beta. So here, this is how they are present alternatively. This is your beta tubulin, that is blue in color, and alpha tubulin, yellow in color. Then blue in color and yellow in color. This is how they are alternatively present. Next comes the actin filament. Actin filament are nothing but the smallest type of filament that is present in the eukaryotic cell, that is about 6 nanometer in size, and they are made up of actin protein. This is how your actin filament looks like, and they are made up of numerous actin proteins put together, and they are about 6 nanometer in size. have the intermediate filament. Unlike the previous one, this intermediate filament is heterogeneous in its composition, made up of several proteins. So we are speaking about the cytoplasm. We spoke about the cytoplasmic membrane, and then comes the function of your cytoplasmic membrane. Here is your cytoplasm, then the cytoplasmic matrix, that is your cytoskeleton. Here, now we are going to learn about the single membrane bound organelles in a typical eukaryotic cell. This includes endoplasmic reticulum, Golgi apparatus, lysosomes, vesicles, and ribosomes. These are the single membrane bound organelles. We are not going to go too much in detail, but we will be learning about the structure and function of these single membrane bound organelles in your eukaryotic cell. Endoplasmic reticulum, as we all know, they are simply divided as rough endoplasmic reticulum and smooth endoplasmic reticulum. As I told before, why we call them rough endoplasmic reticulum is because of its beaded appearance or studded appearance. It has bulging in its surface. That is because of the attachment of your ribosomes to the endoplasmic reticulum. Endoplasmic reticulum is attached to the surface of your ribosomes are attached to the surface of your endoplasmic reticulum, makes it a rough surface, hence called rough endoplasmic reticulum. Where the surface is smooth, it is called as smooth endoplasmic reticulum. These membranes are believed to be attached to the nuclear membrane. If this is the nucleus and this is the nuclear envelope or your nuclear membrane, from there it starts. Always your endoplasmic reticulum is attached to the nucleus. What is the function of your rough endoplasmic reticulum? Yes, it is for the protein synthesis, as it has ribosome attached to it. Smooth endoplasmic reticulum, on the other hand, do not have ribosomes attached to it, but the reason or the ultimate function of your smooth endoplasmic reticulum is the production of phospholipids. These lipids are very important because we know the cell wall of a eukaryotic cell is made up of phospholipids. So continuous production of phospholipids are important. Only then the head and the tail can be produced. That is a phosphate group attached to the lipid molecule. Only then the double layer structure can be formed. How important is protein synthesis? Equally important is the phospholipid production in the cell. Smooth endoplasmic reticulum also transports the protein that is produced by your rough endoplasmic reticulum to Golgi apparatus or your Golgi bodies. Here, the function or ultimate function of your endoplasmic reticulum, your rough endoplasmic reticulum not only produces protein, there are four important functions of your endoplasmic reticulum that we all should know. The first one is protein synthesis, the second one is protein folding, the third one is protein quality control, and the fourth one is protein sorting. It is important to know this because protein doesn't function just the way it is. It needs to be in the proper conformation. For that, proper folding need to be done. Protein correction need to be done. That is undergoing the quality control, and then protein sorting is important. So those four words are mandatory to know: protein synthesis, protein folding, quality control, and protein sorting. Those are the functions of your rough endoplasmic reticulum. Then comes your smooth endoplasmic reticulum. Yes, as I told, it is involved in the production of phospholipids, and also it helps in production of cholesterol. It also helps in the production and secretion of steroid hormones, metabolism of carbohydrates, and a smooth endoplasmic reticulum helps in storing calcium ions. This is the structure and function of your endoplasmic reticulum. Then comes your Golgi apparatus. Your Golgi apparatus or your Golgi bodies is made up of flat disc shaped structure called as Christina. This disc shaped structure, flat disc shaped structure, will is called as Golgi apparatus. As I told you, rough endoplasmic reticulum will produce proteins with the help of ribosomes, that is taken by smooth endoplasmic reticulum, and smooth endoplasmic reticulum gives those protein that is synthesized to Golgi apparatus. They are arranged in parallel to the nucleus, that is in parallel to your endoplasmic reticulum. They are the important site for the formation of glycoproteins and glycolipids. So as I told, they are involved in the protein collection, so they produce or formation of glycoproteins and glycolipids. These Golgi apparatus are eternally present in the eukaryotic cell. They are believed to transport, modify, and package proteins, lipids, and obtain it from the smooth endoplasmic reticulum and deliver it to vesicles. There is always a connection between the organelles present inside the cytoplasm. Protein synthesis from the nucleus, genetic material will give the command which protein has to be produced. That protein is produced by your rough endoplasmic reticulum, that is taken up by your smooth endoplasmic reticulum, given to your Golgi apparatus, so that glycoproteins and glycolipids are produced, and then from the Golgi apparatus it is given to the vesicles. Now the vesicles will go and distribute the proteins in the various parts of the cell, because all these are immotile. Only your vesicles are the motile organelle that can go travel to the diverse part of the cell and deliver the protein thus produced by the ribosomes present in the rough endoplasmic reticulum. If this is clear and the concept would be clear, these secretory proteins and glycolipids, membrane proteins are produced, lysosomal proteins are produced, glycolipids are produced. All these lipids are proteins are produced by the ribosomes and given to smooth endoplasmic, then given to Golgi apparatus, then that is transported to the vesicles, and these vesicles will now go and deliver it wherever required across the cell organelles. So this is how it works, where your nucleus is present, then your endoplasmic reticulum, rough endoplasmic reticulum, then your smooth endoplasmic reticulum will give it to Golgi apparatus, and that will give it to the vesicles. Then next comes the lysosomes. The minute you hear the word lysosomes, it should be like neutralizing or nullifying effect creating organelle. This is a spherical shaped organelle that is filled with hydrolytic enzymes. This has a capability to break down molecules. This is a double membraned organelle. It is made up of, it is a lipid layer, glycosylated membrane proteins are present in the outermost layer. These have an ability to break down cellular waste or debris by engulfing them. And then within them, there is a presence of hydrolytic enzyme, so it is easy to break down these biomolecules. They undergo something called as phagocytosis, where the unwanted cell debris that are there within the cell can be nullified with the help of the hydrolytic enzymes present inside the lysosomes. So they help in breaking down larger polymers, they help in digestion of food, digesting extracellular materials, aiding in the immune response, breaking down owned cellular components, participating in apoptosis, that is a programmed cell death. Lysosomes break down larger molecules such as lipids, carbohydrates, and proteins into their component parts. These helps in reducing the cellular net weight by neutralizing the cell debris present in them. And this is the vesicle, the role of the vesicle. As we know, it takes the protein from the Golgi apparatus and Golgi apparatus and goes and gives it to the various organelles across the cell. This is how it approaches the organelles cell layer. These are the proteins that are present. It goes and attaches first to the target cell layer, and then it digests the tissue or the cell wall, and then the molecules are sent inside. This is how the vesicles do supply the proteins thus obtained from it. And the last one is ribosomes. Yes, they are made up of proteins and RNA. It does not have a membrane, but we are just learning it because it is important in the functioning of all the single membrane bound organelles. It is present attached to the rough endoplasmic reticulum, helps definitely in the production of protein synthesis. Free ribosomes are also making proteins, and that will also be supplied by your Golgi apparatus and thereby by vesicles. Ribosomes is made up of RNA and proteins. These are larger, it is divided as a large subunit and small subunit. For RNA, DNA to convert to RNA, that is transcription, and RNA to protein is your translation, for which these ribosomal subunit attachment to the mRNA is very, very important. This is how it works. Your mRNA has to be attached by your ribosomes. This is the ribosome small unit and the ribosomal large subunit. This has to come and attached to the mRNA, that has to be converted to protein. Under the influence of your tRNA, proteins are produced. Linear chain of amino acids. Many amino acids arranged in a linear chain like this will give protein. So this is what will be happening in the translation process, for which ribosome plays a vital role. So this is all I have for today's video. To quickly give you a recap of what we learned in the single membrane bound organelle, we have endoplasmic reticulum, we have Golgi complex, we have lysosomes, vesicles, and ribosomes. More than understanding the function of each of the organelle, it is important to understand how they work in sync with each other, how the protein production and transportation occurs in the cell, by what is the role of each organelle, and how they work hand in hand is very important. So that is all we have for today's class. So thank you.